One of the most important works we are doing recently is to derive induced stem cells, iPSCs, from patients with a distinct behavior and brain electric activity phenotypes. The reason we want to do that is because there's a really diverse kind of representation and severity among Fragile X patients. And this diversity has contributed to a lot of the complexity in clinical trial design and readout. And some of the clinical trials may actually have run into this complication of data interpretation because of diverse representation of a patient severity and symptoms. So because of that, we decided to derive the iPSC stem cells from the patient with a distinct representation in their severity and the drug response. And this is a collaboration with clinicians and also basic scientists and computation scientists. And our goal is to try to identify, so what are the underlying mechanism for these differences in severity, and can we develop an in vitro model in the cell culture model that we can test some of these drugs before they need to be applied to human clinical trial or patient treatment. So this is really exciting because this is highly relevant to what actually need to be applied to patients in the future, and highly relevant to some of the more pressing and fundamental questions in Fragile X research and treatment.